Hello, this is Gina Piscatelli with a lecture on the gross anatomy, histology, and function of the stomach. So this picture that you're looking at is taken from a cadaver, and it's been bisected sagittally. The esophagus is coming down to enter a certain region of the stomach called the cardia region right here. And then the bulge that comes upward, that's referred to as the fundus. And you can see the folds of the stomach. Right now, the stomach is not distended, and so the folds are visible. But when the stomach fills, those folds stretch out. You can also see the region that narrows. That is called the pyloric region. And there's a valve here called the pyloric sphincter, sometimes called the pyloric valve. That leads into the first part of the small intestine, called the duodenum or duodenum, depending on who's pronouncing it. So in this lecture, we'll review the structure, the gross anatomy, and then we'll look at the tissue types that are found in the stomach and relate those facts to the function of the stomach. So the stomach lies in the upper left quadrant of the abdominal cavity, and it's lined with the peritoneum or the peritoneal membrane. And before we talk too much about the details of the anatomy, I just want to give you some pretty amazing features of the stomach. When the stomach is empty, the lumen is still open enough to have a volume of about 50 milliliters. But after eating, it normally distends to hold up to 1 to 1.5 liters. However, it's able to distend up to 4 liters. That's amazing. I'm sure that wouldn't feel very comfortable, though. So once food leaves the stomach to enter the intestine, it collapses back to the original volume of 50 milliliters. And as I mentioned before, in this diagram, you can see the folds present. Those folds are called rugae. And when full, the stomach loses these rugae because they're stretched out. They're no longer visible. Okay, now some anatomy. There's 4 regions of the stomach. The cardia surrounds the opening from the esophagus. It's outlined in purple there with the word cardia. The dome-shaped part that tucks up against the diaphragm is called the fundus. So this is the diaphragm right here. Put a D there for diaphragm. And the main portion, the middle portion, I guess, is the body, the largest portion. And finally, the fourth portion is called the pylorus, and it's funnel-shaped. And it narrows towards a sphincter made of smooth muscle. And this pyloric sphincter acts as a valve to control how much of the stomach contents enters the small intestine at any one time. And we'll look at that again in a little bit. So in addition to the pyloric sphincter, which I'll review in a minute, there's also another sphincter associated with the stomach. And it's called the cardiac sphincter because it's close to the cardiac region. And it has some other names: gastroesophageal sphincter, meaning the sphincter in between the stomach, gastric, and the esophagus, esophageal. But there are 2 sphincters associated with the esophagus, just the esophagus, not the stomach. And this one is the lower of the 2. So sometimes it's also called the lower esophageal sphincter. I'll call it the cardiac sphincter because we're dealing with just the stomach here. It's composed of smooth muscle, and it relaxes to allow a food bolus that has traveled down the esophagus to enter the stomach. So it has to relax for a food bolus to enter the stomach. The pyloric sphincter is also made up of smooth muscle, and it is normally somewhat open, kind of like you see here. There's a space between the two sides, and it's open enough that liquids and very small substances can get through. But when it closes, it cuts off how much can enter, and that causes a wave of retropulsion, we call it, or a backwards wave of the contents of the stomach. So it closes kind of abruptly and tightly, and so substances are pushed backwards and they can be digested again. Have another go at breaking them down. Okay, now we'll look at stomach histology. Now I'm going to deal first with the mucosa, which is the innermost layer of the entire alimentary canal. In the stomach, the mucosa is composed of simple columnar epithelium. And it's arranged in a pattern that forms pits, shown in the lower right picture. They're called gastric pits. And at the very base of the pit are glands that produce mucus and gastric juice. So gastric glands at the base of a gastric pit make gastric juice. And gastric juice contains enzymes, and it also has a very low pH. The mucus that's produced by the gastric glands protects the simple columnar epithelium from that acidity, that very low pH. The second layer that I'll look at here is called the muscularis externa, and unlike most regions of the alimentary canal, it's composed of 3 layers of smooth muscle rather than just 2. This reflects the mechanical digestion function of the stomach, during which food is pummeled and churned, so mechanical digestion, whereas the gastric glands produce juice that will chemically digest substances. And the extra layer is called the oblique layer. There's always in the alimentary canal a longitudinal layer, meaning muscle fibers that run lengthwise, and a circular layer. The muscle fibers run in a circle around the diameter, or circumference, I should say, of the alimentary canal. But in the stomach, we also have an oblique layer. And I have them labeled here. This is the longitudinal layer with the L on it. And then there's a circular layer. That you'll see all the way from the esophagus down to the intestine, or rectum. In addition, here's the oblique layer that the stomach has. Okay, that's histology. That's all we're going to talk about in terms of histology until we look at the gastric glands in a little bit more detail. So let's now look at the functions of the stomach. The first function is motility, which involves mechanical digestion, breaking down mechanically the bolus, the food bolus that entered the stomach, kind of pummeling and churning the contents. It also, motility refers to propelling the contents towards the small intestine or towards that pyloric sphincter. So the mechanical digestion and the propulsion is a modified form of peristalsis. And as a result of pushing on and kind of kneading like you knead bread, kneading the food bolus. The food bolus now is going to become more liquidy because it gets mixed with the gastric juice and all that mucus, and there's some water in the gastric juice. So now we don't call it a bolus anymore, we call it chyme. C-H-Y-M-E. Now it's this oblique muscular layer that propels the chyme towards that pyloric sphincter. So that's a little entry or introduction into the motility of the stomach. Now let's look at secretion functions of the stomach. Secretion meaning to secrete a substance from the cells, right? So secretion is performed by epithelial cells. And we know already that the gastric glands are going to secrete this mixture of substances called gastric juice. This gastric juice will perform chemical digestion. So it will contain some enzymes and other substances that will break down certain types of nutrients. So there's two components that chemically digest proteins. One component is hydrochloric acid, and that denatures proteins. Then there's pepsin, and pepsin physically breaks down peptide bonds between amino acids. So this is an enzyme that catalyzes a reaction of breaking that bond. So it digests peptides as well. But gastric juice also contains enzymes that break down lipids. For example, gastric lipase. It digests lipids. A little bit minimally, it doesn't do a lot, but lingual lipase, which was in the saliva, has come down with the food bolus, and it's still there. And it is activated by the low pH in the stomach. So you actually have two enzymes that are functioning in the stomach to digest lipids. It's just that one of them has been made up in the salivary glands and one has been made by cells in the stomach. Now there's one more item that is secreted by those epithelial cells of the gastric pits, and this is intrinsic factor. Intrinsic factor is required for erythropoiesis because it helps the small intestine absorb vitamin B12. You actually have to have vitamin B12 in the presence of intrinsic factor to even get it into your bloodstream. And so that is really the main essential function of the stomach that we could not do without. If we had to, for some reason, get rid of our stomach, we could eat, more fluid items, and the small intestine would be able to do some nutrient absorption, but we would have to be administered intrinsic factor to absorb any vitamin B12 and make new red blood cells. Now, another function of the stomach, I guess we can call it, is absorption. However, the stomach really doesn't do any nutrient absorption. So you won't absorb carbohydrates, lipids, or proteins. However, the wall of the stomach absorbs alcohol and aspirin. Okay, let's talk a little bit about the function of motility, gastric motility. Contractions in the stomach occurs in waves, and I mentioned that it's a modified peristalsis, and that's because it does have this rhythmic process where a contraction begins in the cardia or the cardiac region and progresses towards the pylorus. Now, at the same time that this motility is happening, from, we'll say, superior to inferior of the stomach, there's also compression of the contents, right, particularly by the circular muscle. And that will churn the contents. We call it churning. So when you hear your stomach growl, it's usually that gastric motility, which we won't get to talk about too much, what happens when you're hungry and your stomach starts growling. But in general, that's coming from your brain. Your brain is telling your stomach to begin churning because it's time to eat. So the purposes of gastric motility, as I mentioned before, is to mix the food bolus with all those gastric secretions to form chyme, and then to propel the chyme into the small intestine. Now that chyme will be propelled towards the opening, the duodenum, the beginning of the small intestine, and it turns out that about 3 milliliters of chyme squirts into the duodenum at any one time. And the reason only 3 mils goes in at a time is because the pyloric sphincter rhythmically closes. So every once, I don't know what the time frame is, but every 5 seconds or something, it contracts. And so that will ensure that the intestine only receives the amount of chyme that it can neutralize with its bicarbonate ion. And we'll learn more about that later. But the chyme coming into the intestine is acidic, and the intestinal secretions are basic or alkaline to neutralize that acidity. So after a food bolus has arrived in the cardiac region of the stomach, it takes about 4 hours for the stomach to completely enter. And this is a picture that kind of shows you the peristaltic waves that occur in the stomach, starting at the cardiac region. And over time, you see that the wave goes a little bit farther and then a little bit farther yet. And the last picture on the right shows the closing of the pyloric sphincter, and that makes this more liquidy chyme kind of flow backwards. And that just exposes it to another peristaltic wave. And so there'll be more mechanical digestion and mixing with gastric juice, so more chemical digestion as well before it goes back to the pyloric sphincter. Now let's talk about the secretory functions of the stomach. These secretory functions are performed by many different cell types present in the gastric glands. And in different regions of the stomach, the gastric glands have variable numbers of these cell types. Just about everywhere you'll find mucus cells that make secrete mucus to line the interior of the stomach. And probably everywhere you'll have all these other types of cells too, the parietal cell, the chief cell, and enteroendocrine cell. But the type of enteroendocrine cell will vary, and the numbers of parietal cells versus chief cells will vary in different regions. But we're just going to learn the functions of all these and kind of assume they're everywhere in the stomach, which isn't terribly far from the truth. So the mucus cells are closest to the lumen, and that's very important because their purpose is to secrete mucus to protect the lining of the stomach from the hydrochloric acid, which is made by the parietal cells. So the parietal cells are making hydrochloric acid, and the hydrochloric acid will rise and then be kind of sitting on top of the mucus that's already there. The acid is very important for breaking down proteins, as I mentioned previously. But in addition to making HCl, it's these parietal cells that make intrinsic factor. So they're very important for vitamin B12 absorption, which won't happen in the stomach, but the intrinsic factor will go with the chyme into the small intestine, and that's where vitamin B12 will be absorbed. Now the chief cells make pepsinogen. Pepsinogen is an inactive zymogen. Like we saw with clotting factors, they were inactive in the blood. But once the pepsinogen has been secreted, it's now exposed to the hydrochloric acid, and the hydrochloric acid will turn it into an active form, and the active form is called pepsin. And as its name implies, it's going to break down polypeptides by breaking peptide bonds. Now the chief cells also make the gastric lipase, which breaks down lipids. So we've got quite a few things being made by 3 different cell types here. Now we've got the last cell type, the enteroendocrine cell. Entero, I guess, I usually think of that as intestine, but in this case, it just refers to the alimentary canal, right? The digestive tract as a whole. And endocrine refers to the fact that whatever it makes is going to be entering the blood, not entering the lumen of the gastric pit. So the green cells at the base of these gastric glands will release substances that are going to move in the opposite direction. They're going to be secreted this way because that's where, from the out the basal surface of the cells, because that's where the blood is. And endocrine refers to hormones that travel in the bloodstream. So enteroendocrine cells are of 2 different types. One type is called a G cell, and that one makes gastrin. It's a hormone. We'll talk about what it does in a little bit. Or an enteroendocrine cell is an ECL cell. I don't know where that comes from. And that one makes histamine. And usually when you think of histamine, you think of basophils. So this is a new role of histamine. So all of the different components of gastric juice that I just mentioned are listed on this slide. We produce 2 to 3 liters a day of gastric juice. That's a lot. And the pH is usually about 2, but I've put a range here of 1.5 to 3.5. And the purpose of the gastric juice is to chemically digest the bolus, particularly protein and a little bit of lipid. So we have water and mucus, hydrochloric acid, which is going to denature proteins, kill bacteria, and we've learned that it breaks pepsinogen down into an active form called pepsin. Gastric juice is also made up of intrinsic factor and pepsinogen, which gets cleaved to pepsin, so a little bit of pepsinogen and a little bit of pepsin, both, and lipase, gastric lipase in particular, which will digest lipids. So in the mouth we had digestion of carbohydrates and maybe a little bit of lipids. In the stomach we have chemical digestion of proteins and a little bit of lipids. Let's talk about how pepsinogen is converted into its active form a little bit. Okay, so this slide shows that pepsinogen has been released by a chief cell and hydrochloric acid has been released by a parietal cell, and they're both floating around in the gastric juice. When hydrochloric acid encounters pepsinogen, it cleaves part of it off, and that makes it become pepsin. And now it is active, and it can go break down protein. That's what it can do. But what's interesting about this is that pepsinogen is also a protein. So pepsin can break down pepsinogen. So you kind of get a positive feedback mechanism. As pepsinogen is cleaved to pepsin, now any pepsinogen left over that hasn't been converted yet will be acted upon by both hydrochloric acid and by pepsin. So you kind of get this surge in pepsin production. Okay, this slide also might be helpful for you. It shows it points to specific cells and tells you what they release: mucus, hydrochloric acid and intrinsic factor, pepsinogen and gastric lipase, or in the enteroendocrine cells releasing histamine or gastrin, depending on what type they are. So the mucus protects the stomach against acidity because it is alkaline. It contains bicarbonate ions, which, if you remember, the chemical formula is HCO3 negative. It's an anion. And so it can bind with a proton, which is a hydrogen ion. And of course, those parietal cells are busy making a lot of hydrochloric acid, which contains a lot of hydrogen ions. So the mucus is able to neutralize those hydrogen ions. But in addition, the stomach is protected by the fact that epithelial cells are connected by tight junctions. So the hydrochloric acid won't slip in between cells and get out of the stomach, or start, digesting the muscular layer and then the serosal layer. So those tight junctions are really important. There's also a lot of mitosis that happens in the stomach. Those epithelial cells are replaced every 3 to 6 days. So there's stem cells in the lining of the stomach, just to ensure that they stay nice and healthy and form those tight junctions. Okay, we just talked about gastric secretion and the components of gastric juice. And now we want to talk about how is secretion regulated? Does it happen all the time? Does it happen when we eat? When does it happen? And it turns out there's 3 phases involved in the production of gastric juice as well as the regulation of its production. And the 3 phases are called the cephalic phase, the gastric phase, and the intestinal phase. Now there's overlap, like the cephalic phase might not completely be done by the time the gastric phase starts, and likewise the gastric phase might kind of creep into parts of the intestinal phase, but we know that there's specific mechanisms that occur in each one of these phases. So the cephalic phase refers to anything that happens regarding gastric secretion before you even get food into the stomach. Okay, before there's stomach there, any food there. In the gastric phase, now the food bolus has entered the stomach. And the intestinal phase is defined as the period in which chyme first enters the duodenum until, I guess, the stomach is empty. So, gastric secretion begins before food enters the stomach during the cephalic phase. And it is induced by a neural mechanism. So there's neural control. The nervous system is involved. The gastric phase, we will see the continuation of gastric secretion, and there's the nervous system that controls it, and there are also some hormones that keep it going. And then in the intestinal phase, we see that although gastric secretion continues for a little bit right after chyme enters the small intestine, it eventually the stomach needs to be told to stop secreting gastric juice. And there's nervous control during this period, as well as hormonal control that stops gastric secretions. So let's look at the cephalic phase first. During the cephalic phase, the stimulus is the smell of food, taste, sight, the thought of food, probably a little churning. I'm not sure where that comes from except for the brain. And from the brain stem, I should say that first all these stimuli are going to have to be sent to the brain, either through your nose, your eyes, your tongue, whatever. And eventually a signal is sent to your brainstem and leaves the brainstem. The signal leaves the brainstem in the vagus nerve. And the vagus nerve contains parasympathetic motor neurons that stimulate gastric secretion even before the food has been swallowed. It probably stimulates a little bit of churning as well, motility, but it never says that in any of the textbooks. Now let's go to the gastric phase. Now the food bolus has entered the stomach, and the stimulus is mainly the stomach distending. There are stretch receptors in the wall of the stomach. And the response to distension is going to be to increase gastric secretions. Once again, we're going to increase gastric juice production during the gastric phase, which makes sense because the food is right there in the stomach. And there's two neural reflexes that are involved with this, a long reflex and a short reflex. The long reflex involves a sensory neuron in the wall of the stomach connected to that stretch receptor that is signaled to carry an impulse up to the brain. Hey, there's food here now. And then a motor neuron will send a signal down to the wall of the stomach to both contract as well as promote gastric secretions. So that's called a long reflex, and it's how the nervous system participates in promoting gastric juice production. But in addition, when those stretch receptors are activated, because the bolus has entered, and now we've got a little bit of chyme happening too, now there's a short reflex that's kind of instigated. And that will happen entirely by neurons within the wall. So there'll be sensory neurons sending signals to motor neurons located just in the wall of the small intestine, which keeps gastric juice production up as well as motility or muscular contraction. Now the gastric phase is also controlled or partly by hormones. And in this case, we're still going to increase the secretion of gastric juice, but the stimulus is a little bit different. So hormones are going to be released by those enteroendocrine cells, but first they have to be stimulated to do so. And so the stomach will distend. That will help stimulate them, but what really stimulates them is the presence of peptides and the high pH of the food bolus. So before food entered the stomach, it still had a pretty low pH. But as soon as the food bolus enters the stomach, now the pH goes up. And so the response will be overall increased secretion of gastric juice because we need to break down the peptides that just arrived. And in addition, there's a hormone that's going to be produced in response called gastrin, because the enteroendocrine cells will contribute, or will also be stimulated by the neural control, actually, but by the presence of peptides in this case. So the whole purpose in hormonal control is that gastrin, the hormone, is going to enhance protein digestion. And the way that it does so is that gastrin is going to be produced by the enteroendocrine cells, enter the blood, and then travel all over through the blood, and then stimulate those cells in the gastric pits to make more hydrochloric acid and make more histamine. And the reason histamine is so important is because that stimulates even more hydrochloric acid production. So the ultimate action of gastrin right away, this hormone, is to lower pH. That's the first thing that's going to happen. And of course, pH in and of itself, or hydrogen ions I should say, help break down proteins. But in addition, the hydrochloric acid cleaves pepsinogen into pepsin, which also breaks down proteins. You might have heard of gastric acid production causing ulcers in the stomach. And an ulcer is kind of like a breakthrough in the wall, the lining of an organ. Well, it turns out that most gastric ulcers are not due to acid at all. 90% of ulcers are caused by a bacterium that's able to live in the stomach because it's acid resistant. Because it is acid resistant. It is called Helicobacter pylori, and that Helicobacter pylori destroys the mucosa of the stomach. And there's a picture of these little bacteria down here on the bottom. I think they're called helico because they look kind of swirly. Now, some ulcers might be caused by substances other than bacteria that break down the wall, the lining of the stomach. Certain non-steroidal anti-inflammatory drugs could do that because they might reduce mucus production or increase hydrochloric acid production. But these are easily treatable. You can treat them with a histamine blocker, and I'm going to show you why that matters. Histamine will inhibit hydrochloric acid production. But how would you treat 90% of the ulcers? Well, you'd treat for the presence of that bacteria, and so you would give an antibiotic. That's what would work. Okay, let's talk about how hydrochloric acid secretion is stimulated. You know that hydrochloric acid secretion occurs when we think of food, or when the food bolus enters the stomach. And in the neural long reflex, it's due to the parasympathetic nervous system telling these parietal cells to do so, to make HCl. Okay? And the parasympathetic neuron releases acetylcholine. Doesn't matter, it binds to a receptor here, and that causes the parietal cell to dump out hydrogen ions and transport chloride ions into the lumen of the stomach. In addition, we have this gastrin that's been made by a chief cell. But the gastrin, when it was made, not by a parietal cell, but when it's made, it doesn't go into the lumen, it goes into the bloodstream. So this gastrin is traveling around in the blood. And once it reaches the stomach, it can bind to a specific receptor on a parietal cell, which causes the same thing that the acetylcholine did. And that is secretion of hydrochloric acid by the parietal cell. So that's hormonal control of hydrochloric acid secretion. Likewise, when those gastric glands have been stimulated, either by the thought of food or the presence of food, we had some enteroendocrine cells that they're not G cells, they're called ECL cells. I can't remember what that stands for. But what they make is histamine. And histamine in this case is acting like a hormone like gastrin did. It's going to enter the bloodstream, travel to the basal side of this parietal cell, bind to a receptor, and again cause the hydrochloric acid. But the real power of these ECL cells is that some of them don't have to use the bloodstream to get to a neighboring parietal cell. Instead, they can act locally by just diffusing to the neighboring cell, the neighboring parietal cell. So more often than not, histamine functions as a paracrine signaling molecule versus really an endocrine or hormonal signaling molecule. But the important thing for us is there's three things that increase hydrochloric acid secretion: the parasympathetic nervous system, the hormone gastrin, and then we'll just say the signaling molecule histamine. So if you take antihistamines, it will reduce your hydrochloric acid production. Okay, the next thing I wanted to show you is how hydrochloric acid secretion is achieved by the parietal cell. So the orange cell here is a parietal cell. I'll put a big P on it. Okay, and it has these pumps, which is a round red circle. And that pump is called a hydrogen potassium ATPase, or we could just call it a proton pump. That would be okay, because what it does is it pumps protons, also called hydrogen ions, into the stomach lumen. It does so in exchange for potassium, because those are both two cations. So, where does this hydrogen come from? Well, ultimately, I guess you could say that it comes from water that was present either in the blood, in the interstitial fluid, and then eventually in the parietal cell. But the parietal cell can't just take the hydrogen off the water and pump it out. Instead, it needs an acid to be formed. So the hydrogen ions have to be derived from dissociation of an acid. And here's the acid. It's called carbonic acid. You've seen it before with the respiratory system. And this is a product of carbon dioxide binding with the water that's already present in the cytoplasm of this parietal cell. So what happens is venous blood carrying carbon dioxide diffuses into the parietal cell, joins with water, becomes carbonic acid, which dissociates into a hydrogen ion, and that gets pumped into the lumen of the stomach. But another product of breaking down carbonic acid is bicarbonate, this HCO3, which is an anion or negatively charged. So what happens to it? It turns out that it is transported back into the bloodstream. So this venous blood on its way back up to the heart is actually basic. It is more basic because of the presence of this bicarbonate ion, which can bind with any hydrogen ions that happen to be present in the bloodstream. So blood that's leaving the stomach, draining from the stomach, is known as alkaline because of the bicarbonate that has been formed from water and carbon dioxide. So it's often referred to as the alkaline tide. I used this particular picture and slide because I think it's a lot simpler than what's in your textbook. And it basically here says that during digestion, the stomach's HCl production will increase because these pumps are going to pump more hydrochloric acid into the stomach. And that will happen in response to binding of a signal on this side. But a byproduct of that is also bicarbonate production. And bicarbonate production that bicarbonate doesn't go into the lumen, it goes into the blood instead. So blood leaving the stomach is alkaline as a result. And I want to show you the slide or the picture that is present in your textbook. And I've gone through and I've kind of changed the numbers a little bit, but you can walk through it with these numbered steps. So both carbon dioxide, shown here, and chloride ion, they both enter the parietal cell, the basal side of the parietal cell, from the interstitial fluid, but ultimately from the blood. And we know that carbon dioxide can combine with water in the presence of carbonic anhydrase, which is present in these cells, to make carbonic acid. So, after that has happened, now we're going to produce the hydrochloric acid into the lumen. And what are the steps? Well, the first thing that has to happen is the hydrochloric acid has to dissociate, has to break down into bicarbonate and hydrogen. Then, using ATP as a fuel source, this proton pump will pump the hydrogen out into the lumen in exchange for potassium. But the potassium's just going to go right back in there anyway. It just kind of gets used over and over and over again. So now we have hydrogen, and we got the chloride ion just from simple facilitated diffusion into the stomach. Okay. And eventually, this bicarbonate is going to go back into the bloodstream. So that's the picture that's in your textbook. Okay, the last phase that regulates gastric secretion happens when the chyme enters the intestine. So this is called the intestinal phase. And there's both neural control and hormonal control. So first, let's do the neural control. What happens in the intestinal phase is the chyme that enters the first part of the intestine, called the duodenum, the word's right here, duodenum, is acidic, has a low pH, and it's got some fats in it, okay, because they haven't been broken down completely. And the nervous system will be stimulated, and so sensory information will go up to the brain, and then a sympathetic nerve will respond to inhibit gastric secretion. Now, why do we want gastric secretion and motility to be inhibited at this stage? Because we need to protect the small intestine from any further acidity. And if the stomach is already propelling the contents into the intestine, then it's pretty well digested it as much as it possibly can. So I just described the long reflex, which involves the sympathetic nervous system output. But as before, any neural regulation in the digestive tract will also have a short reflex component, because the digestive system has this enteric nervous system in its wall. So there's also something called an enterogastric reflex. But the same thing happens. So in response to the sensory neuron in the duodenum sensing the presence of an acidic fatty chyme and distension, the sensory neuron will actually signal some of the enteric motor neurons as well, which will again inhibit gastric secretion. So there's nervous control. Once the chyme enters the duodenum, the nervous system helps stop gastric secretion. But there's also hormones involved in stopping gastric secretions. We're still in the intestinal phase, right? So the chyme has entered the duodenum, and in response to the low pH now present in the duodenum, low pH of the chyme, there are some endocrine cells lining the duodenum that make a hormone called secretin. And secretin inhibits gastric secretion. Now, also the chyme has some lipids that weren't fully digested. And there are some enteroendocrine cells in the duodenum that respond to the lipids. And those enteroendocrine cells make cholecystokinin. Chole, I think, refers to the gallbladder. So does cyst. I don't know what kinin means. But cholecystokinin refers to the fact that it's going to have something to do with the gallbladder later. And in response to lipids, it's going to inhibit gastric secretion as well. So here's a picture that shows a little bit more anatomy related stuff on how hormones would work with the digestive system. So first, just notice that we've got liquefied kind of chyme that's been squirted into this first part called the duodenum, the first part of the small intestine. Now, some of the enteroendocrine cells are going to make secretin, and that's yellow. Okay, and now the secretin is in the bloodstream. So secretin can affect many organs. We're looking now at how secretin affects the stomach when it travels in the bloodstream back to the stomach. It inhibits motility and secretion, gastric juice secretion. But we're going to see that later secretin is going to have an effect on the pancreas as well. And this picture's got the pancreas in it and the liver. But anyway, also the chyme is very low in pH, and it was the low pH that triggered the secretin production. But the chyme also has lipids in it, and that's going to cause some of the cells to make cholecystokinin. And in this picture, cholecystokinin is red. And so that hormone enters the bloodstream, travels all over the place. It can affect several organs. It affects the liver, affects the gallbladder, but right now, and the pancreas. But what we're looking at right now is cholecystokinin affecting the stomach. And it does so by inhibiting gastric juice secretion. Hope I didn't make that too confusing for you. So I want to stress that it's important that you know that secretin is made by the small intestine in response to the duodenum detecting low pH of chyme. And it's also important for you to know that the small intestine makes another hormone called cholecystokinin when lipids are detected in the duodenum. Right now, all you have to know for their actions is that those two hormones will inhibit gastric secretion. Okay? So what we've seen so far in our discussion of gastric secretion regulation are some things that stimulate the production of gastric juice and some things that inhibit the production of gastric juice. So what stimulates it? Well, the thought of food, aroma, taste, sight of food, probably hunger, right? All mediated by the parasympathetic nervous system. That's going to increase gastric juice production. Also, distension of the stomach cause some neural reflexes. High pH in the stomach because the food bolus that came in stimulated gastric juice production. The presence of peptides in the stomach. And then some endocrine signals from the gastrin and paracrine signals from histamine specifically increase the hydrochloric acid content of the gastric juice. Now, what stops gastric juice secretion? Well, lowering sympathetic output would, but in actuality, it's the sympathetic nervous system putting the brakes on the parasympathetic that does it. So the sympathetic nervous system will inhibit gastric motility, inhibit the parasympathetic nervous system, and generally inhibit the gastric glands from secreting anything. Another thing that does is distension of the duodenum. So when the chyme begins entering the small intestine, that begins to stop gastric juice production. Acidity and lipids in the duodenum will cause the release of these hormones, which decrease gastric juice production. Thank you very much. That's the end of this lecture.